Modern immunology defines systemic lupus erythematosus as a spatially organized, self-amplifying failure of immune tolerance. While autoantibodies are a hallmark of the disease, they represent a downstream output of deeper, interconnected biological circuits. The disease mechanism rests on four specific pillars: innate sensing, interferon amplification, adaptive cellular collaboration, and the survival of long-lived plasma cells. Visualizing SLE as a system of closed loops explains why the disease can persist and adapt despite standard treatments. Researchers have identified over 150 susceptibility loci across the human genome that contribute to SLE. These genetic variations do not trigger the disease directly, but instead modify immune thresholds, apoptosis rates, and tolerance checkpoints. External factors like UV radiation, Epstein-Barr virus, and SARS-CoV-2 place physical stress on these genetically sensitized systems. These triggers induce epigenetic modifications, such as DNA hypomethylation in T cells, which lock autoreactive genes into an active state. According to the theory of self-organized criticality, repeated immune stimulation eventually pushes these circuits past a point of stability. This convergence of polygenic risk and environmental stress lowers the tolerance threshold, priming the system for a systemic collapse of regulation. Healthy physiology relies on the rapid, silent clearance of apoptotic cells by macrophages and complement proteins. In systemic lupus erythematosus, specific genetic deficiencies in critical molecules like C1q and DNase1 cause this essential clearance process to completely fail. This failure leaves intracellular debris, including nucleosomes, double-stranded DNA, and RNA, exposed in the extracellular space. The innate immune system mistakes these self-nucleic acids for viral pathogens, triggering an inflammatory response against the host's own genetic material. These antigens form immune complexes that are actively internalized into the endosomes of plasmacyted dendritic cells. This accumulation of cellular waste provides the persistent fuel that drives the innate immune engine forward. Inside the cell, endosomal receptors TLR7 and TLR9 bind to the internalized self-RNA and DNA. This binding recruits the MyD88 adapter protein to initiate the signaling relay. The signal then propagates downward through the IRAK4 kinase. IRAK4 activates the TRAF6 complex next. A secondary split activates NF-kappa B, driving inflammatory cytokine production. The primary signal reaches master transcription factor IRF7. Activated IRF7 translocates into the nucleus to induce transcription of type 1 interferon genes. This rapid molecular relay converts a localized sensing error into a self-sustaining systemic inflammatory state. The plasmacytoid dendritic cells rapidly release a massive, destructive wave of type 1 interferon, primarily IFN-alpha, throughout the entire biological system. This interferon binds to the IFNAR receptor on surrounding immune cells, activating the JAK-STAT and TYK2 pathways. This induces the interferon signature, a widespread change in gene expression that shifts immune cells toward an autoreactive phenotype. In the lymph nodes, this environment supports the development of pathogenic DOCK8 T follicular helper cells. These rogue TFH cells utilize the CXCR5 receptor to migrate into germinal centers, bypassing normal tolerance checks. Using the cytokine IL-21, these TFH cells drive autoreactive B cells to undergo affinity maturation and hypermutation. The innate interferon storm provides the environmental pressure necessary to wire the adaptive immune system for chronic autoantibody production. B cells differentiate into short-lived plasmablasts that correlate with acute flares, and long-lived plasma cells that migrate to specialized niches. These long-lived plasma cells survive CD20-targeted therapies like rituximab. Their survival instead depends on the BCMA receptor. The cytokines BAFF and APRIL provide the signals necessary to rescue these autoreactive cells from natural deletion. Simultaneously, abnormal neutrophils undergo a violent process called NETosis, extruding sticky DNA webs that trap antigens and further activate plasmacytoid dendritic cells. Immune complexes deposit in organs like the kidney, creating localized inflammatory microenvironments. In lupus nephritis, VCAM1-positive epithelial cells and TGF-beta-driven myofibroblasts actively restructure the tissue architecture. These spatially organized microenvironments drive chronic fibrosis and actively prevent physiological tissue repair. This cyclical model links genetic susceptibility directly to end-stage fibrotic niches. Innate activation through the interferon axis drives TFH and B-cell collaboration. In turn, tissue destruction feeds fresh antigens back into the innate sensing phase, completing the loop. This self-amplifying circuit of nucleic acid sensing, interferon signaling, and cellular help maintains the chronic state of the disease. Modern treatment protocols have moved toward the precision blockade of these specific network nodes. Hydroxychloroquine serves as a foundational therapy by directly inhibiting the endosomal TLR7 and TLR9 receptors. Higher up, anifilumab blocks the IFNAR receptor to neutralize the type 1 interferon axis. Belimumab and telitacicept target B-cell survival signals from BATH and APRIL. To address chronicity, BCMA CAR-T therapy eliminates the long-lived plasma cells resisting standard treatments. This integrated framework provides the biological rationale for combining targeted therapies to break the inflammatory cycle and achieve clinical remission.